يعني كثير موبايل مرتبط بالدي ان اي برضه فلوريسنت. اوكي. So all of the techniques are USA ولكن طريقة الديتكشن بتكون على سبيل المثال radiation, color بالانزيم, fluorescent تألق بسموه, chemiluminescence اللي هو الضوء, كل هذول عبارة عن detection بسموهم signals. هلا كيف بنكشف عن هو سلايد واحد بس الفكرة انه في عندنا هون انا راح اكتب لعبد الرحمن المفروض
The LISA enzyme-linked immunosorbent assay. The LISA enzyme-linked immunosorbent assay is a plate-based assay technique designed for
نيجي على مثلا حسب الترتيب الموجود عندنا اللي هو ريد المناسب.
detecting and quantifying antibodies, proteins, peptides, and hormones. The LISA tests
Hi, and welcome to my video series biotechnique.
are broken into several types. The LISA enzyme-linked immunosorbent assay is a based assay technique designed for detecting and quantifying substances such as antibodies, proteins, peptides, and hormones. The LISA tests are broken into several types. The LISA tests are broken into several types. In this video we'll talk about indirect ELISA. And we are going to see how it can be applied for HIV virus detection. When a person is infected with the HIV virus, the immune system creates proteins called antibodies as part of the body's natural response to fight the infection and to neutralize the pathogen. Usually, after an incubation period, the body begins making antibodies that are detectable for months and even years. The indirect ELISA test is used to detect the presence of these antibodies. First, a blood collection procedure is performed by venipuncture. Blood is the preferred specimen for testing because it has a higher concentration of HIV antibodies than oral fluid. After blood collection, the next step in the process of HIV testing is sample preparation. The samples are carefully placed into a centrifuge machine. During centrifugation, the blood components separate based on their density. After the centrifugation process is complete, the serum layer is carefully removed from the tube. The serum is then transferred into a new properly labeled tube for further testing. Finally, the samples are appropriately stored in a designated area until they are ready for analysis. Once the serum samples are prepared, the next step is coating the plate with the antigen of interest. In this step, plates of 96 wells or strips of 8 wells are used. The well strip is placed into a support frame. Then, an antigen solution is added. The HIV virus has a complex structure consisting of various proteins, including GP120, GP41, and P24. To detect HIV antibodies, one or more of these antigens can be selected as the specific antigen. The selected antigen solution is carefully added to each well. Then, the well strip is covered with adhesive plastic and incubated at room temperature or in a laboratory oven. In ELISA, the most commonly used material, as the solid phase, is polystyrene. The antigen is immobilized onto the polystyrene surface. Next, the adhesive plastic is
بسموها ادزوربشن. ادزوربشن معناها بلزق. انا بدي اثبت هذول المولكيولز وين؟ بالول. ليش هذول المولكيولز تحديدا؟ لانه بالنسبه له بدور على الانتي بودي تبعهم. بشوف الموجود بالعينه ولا لا. واحد من
removed. Then the well strip is overturned and tapped to remove the excess antigen solution. Also, an absorbent paper towel is used to ensure the thorough removal of the antigen solution. After the excess antigen solution has been removed, the next step is to wash the wells with a wash buffer. The wash buffer is a specially formulated solution used to rinse the wells and remove any unbound substances. By using a wash buffer, we eliminate any unbound antigens. Once the wash buffer has been discarded, an absorbent paper towel is used to remove any remaining liquid. The next step is to block any remaining unoccupied sites on the solid phase. During this step, a blocking solution is used, which typically contains proteins such as BSA, serum, non-fat dry milk, or casein. Blocking solution is added to each well, ensuring complete coverage. The well strip is then covered and incubated. The proteins in the blocking solution create a barrier on the plate, preventing the target antibody from binding to these sites in subsequent steps. Once the blocking step is complete, the adhesive plastic is removed. Then the well strip is overturned, allowing the blocking
هلا اللي بدنا نضيف بعد شوي هنا انتي بادي صح؟ الانتي بادي عباره عن ايش؟
بروتين.
هلا زي البروتين هذاك ممكن يلزق بالسيرفس. حتى احمي البايندينج وما يرتبطش بالسيرفس نستخدم شيء اسمه بوفاين سيرم البومين. عارفين شو يعني بوفاين سيرم البومين؟ الثور تبع البوفاين اخذوا منه السيرم واخذوا الالبومين اللي فيه والبروتين. حضروا منه باودر بنباع بالاسواق. هذا بنستخدمه كبلوكينج. ايش يعني بلوكينج؟ بغطي الاماكن اللي ممكن العينه تبعك تلزق فيها. مشان اضمن انه بس المتوفر الان الانتيجين او البروتين اللي انا بدي ارتبط فيه او بدي اعمل له
solution to drain out. Next, the absorbent towel effectively absorbs any remaining solution. After removing the excess blocking solution, the wells are thoroughly washed with the wash buffer. This step helps to ensure that any remaining blocking protein is thoroughly removed from the wells. After discarding the wash buffer, any residual liquid is removed using an absorbent paper towel. After the blocking step, the next step is incubation with the samples. The serum samples obtained from patients are added into the wells, ensuring that each well receives the appropriate sample. After the serum samples have been added, it is important to include negative and positive controls to evaluate the test. The well strip is then covered to create a control
ايش بيعمل الكنترول دكتور؟ مش فاهم ايش الكنترول بيعمل؟
الكنترول عينه معروفه معمول عليها كواليتي اشورنس بالمختبر انها هي نيجاتيف. ممكن تكون بس عباره عن صلاين ملح ومي او قفل. عين ثانيه فور شور فيها انتي بودي للاتش اي في متاكدين منه. ليش بستخدم واحده نيجاتيف وواحده بوزيتيف؟ عشان اتاكد انه النيجاتيف تبعتي اللي ما فيهاش كعينه اتس ترو نيجاتيف والبوزيتيف از ترو بوزيتيف لانه النيجاتيف والبوزيتيف الكنترول اللي انا بعرفه is working. فهي بنسميها quality assurance, quality control على الشغل. يعني ما بصير اشتغل بدون كنترولز. تمام؟ لانه اذا طلع بوزيتيف you don't know بوزيتيف لانه البروسيجر غلط ولا صح. شفت خطوات الواشنج هذول؟ جزء واحد ما عملش واشنج. او ما عملش بروبر واشنج مثلا.
يعني مشكلة كاملة.
ممكن يطلع لك فيها فرصة هذا يعني جزء من العمل اليومي بالمختبر انك تعمل كنترول على الشغل.
controlled environment for the incubation process. During the incubation period, the antibodies recognize and bind specifically to the corresponding antigens that have been immobilized on the surface of the wells. Once the specific recognition between the antibody and the antigen has taken place, the next step is to discard the sample solutions from the wells. Next, wash buffer is added to the wells. The wash buffer helps leaving only the desired antibody-antigen complexes.
فمثل الهرمون هو اللي ماسك ندور عليه.
After washing away any unbound substances, the next step is the incubation with a conjugated antibody, also known as a secondary antibody. By using a conjugated antibody, we can detect and quantify the presence of the targeted antibodies.
Conjugated with enzyme.
The conjugated antibody solution is added to each well, then the well strip is covered and incubated.
يعني اول ما اسمع
The-
يعني كيف بدي اربط انتي بانتيجين بانزيم او ببروتين او بفانكشنال جروب، هاي صارت يعني عباره عن سيلي ثينكس كل يوم بنعمل بالمختبر، عباره عن كيميكال رياكشن تربط انزيم مع الانتي بس. طيب ليش بدي اربطهم؟
Conjugated antibody is specifically designed to bind to the antibody-antigen complexes present in the wells. Next, the conjugated antibody solution is removed from each well, ensuring that any remaining liquid is eliminated. After using the conjugated antibody, the next crucial step is to perform a final wash to remove any excess conjugated antibody. The wash buffer helps to wash away any remaining antigen complexes. After the final wash, the next critical step is the detection of the antibody-antigen.
المادة اللي بتأكتف الإنزيم
بتاكتف
أنا بتبين الإنزيم بالضبط
شو بمثل تبين انزيم؟
صبغة ممكن
سبستريت انسان فالديتكشن سبستريت.
complexes. This is achieved by using a substrate that initiates a reaction, resulting in a detectable signal. The substrate solution is carefully added to each well. Then the well strip is covered and incubated. To enable detection, the secondary antibody is typically labeled with an enzyme, such as horseradish peroxidase, HRP.
عارفين شو الهورس راديش بيروكسيديز؟ عصير فجل بالمعنى العام. فجل هذا بالمعنى البسيط يعني نحكي بلغه عاميه. الهورس راديش عباره عن نوع من انواع الفجل. اوكي؟ بيعملوا اكستراكشن للبيروكسيديز انزيم موجود فيه. تعرفوا طعمه الحروره اللي في البصل احيانا او في سوري بالفجل هذا البيروكسيديز انزيم. رايت؟ وبالتالي تقدر تعمل اكستراكشن لهذا الانزيم او هذا الانزيم من الهورس راديش بلانت على سبيل المثال او من اني اذر بلانت. بالتالي اللي بدي اوصله ليش بحكي هاي المعلومه؟ انه الساينس يعني sometimes looks complicated بس based على اشياء يعني trivial سهل يعني متناول اليد. لما قلت بوفاين سيرم البومين اسم معقد بس هو عباره عن بروتين اخذنا بدل ما ناخذ من الهيومن اخذنا من البوفاين لانه سهل كميات كبيره. طلعوا الالبومين اللي فيه وبعدين عملوا له لايفلايزيشن باودر وبيبيعوه بالاسواق بس بيكون بيور طبعا معالج. نفس الشيء هذا الانزيم بدك تعمل له اكستراكشن يعني كاركترايزيشن تربطه على انتي بودي فكل هذا يو كان دو ات. هذا سبستريت.
Various detection reagents have been developed for HRP, but one of the most widely used chromogenic substrates is tetramethylbenzidine, TMB. In the presence of hydrogen peroxide, HRP enzyme catalyzes the oxidation of TMB, resulting
على الار اس راديكال فري راديكال
لما يكون في عندنا سبستريت زي هذا مع الهيدروجين بيروكسايد
in the formation of two intermediate oxidation state products. One product is a colorless TMB cation radical, which is in equilibrium with a blue-green color.
الفرق بينهم conjugation تمام and they will be colored بعطي اللون.
Charge transfer complex, CTC. When antibodies are present, a color change occurs, and the wells exhibit a blue color.
كل السوليوشن بتغير لونه.
Next, prior to photometric detection, the reaction is commonly stopped by lowering the pH of the reaction mixture using a strong acid, such as sulfuric acid.
اوقف الرياكشن مش يضل شغال يعطي لون كل سنه.
Following a second 1-electron oxidation event facilitated by HRP, the blue-colored TMB product is transformed into a yellow-colored diimine oxidation product. The addition of the acid halts the enzymatic reaction and stabilizes the color.
بضيف اسيد، الاسيد بيحول هذا اللون الازرق الاصلي الى لون اصفر بروبورشنال. قديش عندك ازرق بتحول الى اصفر. السبب further reduction by acid. تمام؟
Development for subsequent measurement. The color change from blue to yellow is indicative of a positive result in the ELISA assay. Next, for confirmation
بتشوف لون اصفر، لون اصفر هذول لون ازرق لسه اللي هو الاوريجينال كلر. هلا بنحطهم بريلا.
and interpretation of the obtained results, the spectrometer instrument is used to read the absorbance in each well and to confirm whether the patient is carrying the HIV virus or not.
طبعا الابزوربر شو يعني؟ عباره عن مصدر ضوء بمر بالعينه. الان هذا اللون الاصفر بيمتص درجه معينه، ويف لينث معين. بالتالي الامتصاص يعتمد، درجه الامتصاص تعتمد على التركيز. تمام؟ وبعطيني اياها شكل رقم. كل ما كان الامتصاص اعلى الرقم اعلى، بالتالي عندي كونسنتريشن اعلى. هيك بنقيس الكونسنتريشن لاي شغله. الاتش اي في لان هذا الفيديو كان يعني بين ايدي
دكتور طب ليش اخذ ثنتين تو سبيسيمن؟ ليش اخذ عينتين يعني في البدايه مش عينه واحده؟
عشان واحد يوزن واحد يعيش واحد
عشان يفرجيك بس مش ثاني.
يعني ما فيش سبب.
لا لا بالعاده زمان كان البليت كامل 96 عين فيخرب البليت. الان من 10 سنوات تقريبا اكثر صار يعطيك ستريبس تركبهم 8 مشان توفر، لانه تخسر 96 ول مشكله كانت.
Hi, and welcome to my video series of biotechniques explained in 5 minutes, where I explain a biotechnique in less than 5 minutes. Hit the subscribe button. Which uses antibodies to detect and quantitate the amount of antigen in a sample. Here is a multi-well plate where you would add your sample that need to be analyzed and where you want to know about its concentration. So let's look at one well to understand the principle a bit more details. So let's say you want to detect an antigen A, and you ask the question, this antigen A is present in the patient sample or not. So first you coat the well with antibody against A, or anti-A let's call it, and now you put radio labeled antigen A, or let's say any other kind of labeled antigen A, and lastly you put patient serum derived or patient serum or patient's body fluid which expected to have antigen A. And that's the question, whether the patient serum has antigen A or not. That's the thing that you want to.
unlabeled antigen هذا وين موجود بالعين. في عنده labeled 1 اللي هو بعطيه راديو اكتيفيتي، اللي هو عندك الانتيجين نفسه لكن معمول له راديو ليبل. في عندك الانتيجين او الهرمون اللي تدور عليه، هذا هرمون ليبل، هرمون بالعين. الاثنين حطيناهم على نفس الانتي بودي، راح يصير كومبيتيشن، رايت؟ منافسه. فالراديو ليبلينج كل ما زاد، شو بكون تركيز العينه او تركيز الهرمون؟
اقل.
تختلف عن الإلايزا شوي.
detect. So simply, radioimmunoassay relies on the principle of competitive binding, where a radioactive antigen, this is a tracer, would actually compete with a non-radioactive antigen to bind to this particular antibody shown here. Now, if the patient sample has huge amount of these unlabeled antibodies,
Just competition binding or competitive binding between antigen اللي already طبعا هذه الشركات بتحطه راديو ليبل، بس المشكله في هاي التكنيك انها مش ستيف، هاي كانت ديس ادفانتج. بس الادفانتج تبعتها انها سنسيتيف، هايلي سنسيتيف، لانه الراديو ديتكتور السنسيتيف تبعته عالي بالراديشن.
antigens, then radioactivity would be decreased progressively with the increase of the antigen concentration. And a nice plot could be derived from that, which is a linear regression. From this, by extrapolating this plot, we can understand the concentration of the patient's sample where the
معينه مثلا تعمل ستاندرد كير، في عندك عينات معروفه التراكيز، تمام؟ تعملهم ميجرمنت، بتجيب العينه تبعتك وبتقيس الراديشن مع البروسيس العادي، ونفرض انه الراديشن طلع هون كقيمه، تعملها اسقاط على ستاندرد كير and you will get the concentration proportion. طبعا لها يعني
antigen is present, and based on the standard curve we can get accurate value of the concentration. So if you have a higher concentration, radioactivity would be very low, or radioactivity or the rate of decrease of radioactivity would be quite high. And if you have a low level of antigen in the patient sample, radioactivity won't fall that much. So depending upon how much percentage of the radioactivity is reduced gives us a quantitative measure about the amount of antigen present. Now let's say we have a patient scenario of 1 and a patient scenario 2, and both the cases, as you can see, patient 2 has more antigen present in its serum. So as expected, after washing these things, what you would see is the second case is the radioactivity would really fall, because there most of the labeled antigens are replaced by unlabeled antigen. Why is so? Because unlabeled antigen was higher in concentration, whereas in the patient 1 the unlabeled antigen was not so much. As a result, not too much of radioactivity is going down, and there are few radioactive antigens which are still labeling the antibody. So based on these observations,
از سمبل نفس الشيء هذا إلايزا ولكن بتكنيك مختلفة. So you know the concept now. في عندنا الفلوريسنت بس.
Hello friends, welcome to BMH Learning. This video is about fluorescence polarization immunoassay. The basis of the assay is fluorescence anisotropy, also known as fluorescence polarization. In fluorescence polarization immunoassay, polarized light excites a fluorescent label. As this fluorescent label is bound, the degree of polarization of the fluorescent emission from the label increases. FPIA combines two technologies to determine analyte concentration. First is competitive protein binding, and the second is fluorescence polarization. Competitive binding requires two antigen or analyte systems: analyte from the sample and tracer labeled analyte provided by the assay reagents. The analyte from the sample competes with the analyte tracer for binding sites on the antibody. Each analyte system attaches to binding sites based on its relative concentration. If the sample contains a high concentration of the analyte, then many analyte tracer molecules are left unbound. When excited by a polarized vertical light, these small fluorescent molecules rotate rapidly, emitting light in many different planes. The result is a decrease in polarization. If the sample contains a low concentration or no concentration of the analyte, then the analyte tracer binds to the antibodies, resulting in few unbound analyte tracer molecules. These large molecules, that is, complexes of analyte tracer bound to antibody, rotate slower. Rotation is slowed enough so that light is emitted in a single plane. The result is an increase in polarization. This was all. Thanks for watching.
الفرق بينهم بسيط. هلا هون في عندي المادة اللي بدور عليها اللون البينك هرمون.
Hello friends, welcome to BMH Learning. In a single
الانتيبوديز اخذ العينه ما اخذش البولاريزيشن ايجنت، فصار في عندنا ريدكشن في البولاريزيشن decreased and the free tracer rotation is fast. هذول شو بدوروا بسرعه اعلى المولكيولز، وبالتالي بعطيك اشاره اقوى. تمام؟ بالنسبه polarization means low sample. High polarization means high sample concentration. It's competitive binding. بس البولاريزيشن هون هو الفلوريسنت polarization مرتبط مع تركيز العين. كل ما كان اعلى تركيز العين كل ما كان polarization
أكثر راديو أكتيف.
Hello friends, welcome to BMH Learning. This video is regarding chemiluminescence immunoassay. Chemiluminescence is defined as the emission of electromagnetic radiation caused by a chemical reaction to produce light. CLIA is an assay that combines chemiluminescence technique with immunochemical reactions. Depending on different luminescence mechanisms, CLIA includes direct chemiluminescence immunoassay, chemiluminescence enzyme immunoassay, and electrochemiluminescence immunoassay. Principle: the assay involves use of stationary solid particles coated either with the antigen or antibody of interest. Let's say we are using stationary solid particles pre-coated with antibody. In the presence of complementary antigen in the sample, the paratop of the antibody binds to the epitope of the antigen to form an antigen antibody or an immune complex. If we are doing it for chemiluminescence enzyme immunoassay, then an enzyme such as horseradish peroxidase is first used to label an antigen or antibody after immunoreaction. Post-incubation or immunoreaction, which ensures intact immune complexes are formed, the substrate is added.
horseradish peroxidase بس هون شو استخدم؟ مادة تعطي الضوء بدل ما تعطي لون. هذه المادة
Luminol as a luminescent substrate emits light by oxidation reduction reaction under the action of the enzyme labeled immune reactant and the luminescent reagent. The light intensity is directly proportional to the amount of labeled complexity.
ليش الانتيجين انالايت؟ ايش معنى انالايت؟
كإكزامبل الانالايت بيكون هرمون بروتين.
ايش معنى انالايت؟
ايش؟
انالايت ايش معنى؟
انالايت معناها المادة التي ستحل يتم تحليلها مخبريا.
اوكي